Thanks, Philip, and thanks, Bartosz, for this nice introduction into what this partnership is actually about between LUMICKS and Spektrometria. And I, in my role as an application scientist, will now dive a little bit more into the scientific or technical details of this technology as such that we call dynamic single molecule analysis, which we think is a very useful and unique tool to dissect biomolecular interactions. And after having introduced this technology as a tool, I would like to dive a little bit into some real-world scientific applications where this technology has been used to study biological processes ranging from studying gene editing errors all the way down to how cytoskeletal filaments interact with each other. Let's start with the background information. So typically, the hypothesis of a molecular mechanism cannot be proven directly with existing methods in structural and molecular biology. What I mean with that is the following. Imagine you have a molecule such as a helicase, and you know that this helicase is teaming up with some cofactor, and you know it's directionally opening up DNA. But it's not really clear how this happens. It could either be that the protein binds to DNA, it lands there, it diffuses back and forth. At some point, it already it encounters an already opened part of the DNA and then binds the cofactor and really starts to directionally open the DNA. And it could also be that already in solution in the cytoplasm or in the nucleoplasm, this helicase binds the cofactor here in red, and then as a complex binds to DNA, it diffuses back and forth, and then upon an additional stimulus, for example, ATP, ATP, this complex then starts to, as I said before, directionally open up DNA. As I said before, it's not really clear how this could happen in the real world. And we think that you need three things or three aspects of a tool that could allow you to really distinguish different molecular pathways. The first thing is the following. Actually, it would be really nice to be able to directly visualize the location and dynamics of individual proteins. Just imagine that you have the ability to hold a single DNA molecule and observe how a certain protein binds there and diffuses back and forth. The second aspect is that it would be amazing to have control over the stepwise assembly of such a biological complex. In principle, you want to expose your DNA and the complex itself to different environmental conditions with different additional components and see how it builds up step by step and how it at some point becomes active and what happens if you add even more additional components. The third aspect I would like to talk about is it would be also great to be able to modulate the molecular system internally and also externally. For example, by pulling the DNA and see what, for example, mechanical tension does to your process, or by exposing your complex to different drugs that are supposed to modulate the action of this complex. And having all these three things at hand, being able to directly visualize individual protein dynamics on individual DNA molecules, being able to sequentially assemble these complexes and being able to modulate the external conditions, we then think that you can really understand molecular mechanisms at the very bottom of the scientific truth. And how we implement this idea is basically in the following way. We call it the C-trap, and it's a combination of optical tweezers with fluorescence, or if you wish, label-free microscopy. How it looks is the following. So it's this beautiful black box, which has three major components. It has microfluidics, it has an imaging module, which is typically a confocal fluorescence imaging module, and it has these optical traps. Now I will now go step by step through these different components and explain what you can do with them. The first component I would like to talk about, and this is probably the least familiar to most of you, is optical trapping. So that's nothing scary. It's basically just a focused laser beam that helps you to trap, for example, small plastic particles, and not only trap them and manipulate and move them around, but they also allow you to measure forces that act on these particles. Now you might be wondering, okay, why do I need to trap plastic particles? In principle, you need them as handlers. So you can trap functionalized plastic particles. In that case, you see two of them trapped, connected with a DNA molecule. And now combining this trapping capability with fluorescence imaging, you really can see how different biomolecules, different proteins of different colors bind and unbind to and from DNA that is here in between these two beads. right? One bead here on the left side trapped, the right bead also trapped in a different trap, and then connected by this DNA. And on this DNA, you can of course see how really nicely these proteins bind and unbind and diffuse and replace each other. The third technical part I would like to talk about is the microfluidics. So this is basically a glass chip, a small microscopic glass chip that is connected to different liquids, and they flow side by side next to each other without a real, let's say, physical boundary. They just flow next to each other in a laminar flow field, and they basically form the basis of your experiment. So what you can now have is, for example, you have one channel with a solution that has plastic beads inside. You first go here with your two optical traps. You capture two plastic particles. Then you go to the next channel, which is just close by. So basically within a part of a second, you can go to the next channel, catch a DNA molecule that can specifically bind to your beads. This is typically done by beads that have stratavidin coding and biotin-related DNA. And now you can go with this dumbbell construction here into this channel that has the fluorescently labeled, in that case, blue proteins, and you can observe how these proteins bind and unbind to your DNA. And then you go to the next channel that has a different protein that might interact with your first protein, and then see how this works. And then you can also move this whole construct, the two beads with the DNA and the proteins, to a different channel that, for example, contains a certain drug, and see how this drug modulates the action of these proteins on this single DNA molecule. To see this whole experiment in action, we've prepared this small example experiment here. Again, what I've shown before was these two optically trapped beads connected with a DNA molecule. It's a little bit under tension, and it's already a little bit opened here. So here you have a so-called melting bubble, and you already have a fluorescently labeled protein here. And what we will now do is we will expose this protein to different environmental conditions using the fluorescence imaging system and also the microfluidic system. And we will monitor, basically we will scan along this DNA molecule and see what happens there. And this generates a so-called chymograph, which is basically a history of what's happening on your DNA. So you have the fluorescence scan, and then you generate lots of these scans over time, and you plot what's actually happening on your DNA. Let's see what happens. So here and here you have the position of your beads, and in between you have the DNA. So now what's happening is you scan along your DNA and you see that this protein that is already bound to the DNA is diffusing back and forth. Now you can expose your DNA to a different protein that is binding to single-stranded DNA, and you see how already in the fluorescence scan, you see how this blue protein in that case codes the single-stranded part of the DNA. And then once the helicase encounters the opening fork and also binds this additional factor, because we're moving it to a different channel that has this additional factor, you see how it becomes active. So it's opening up DNA, and then we can pull it. We can destabilize the DNA by mechanically pulling, and we can speed this process up. And then we can move this whole DNA construct into a channel that does not have ATP anymore. And since this is an active process, it will stop. And now what we have here is this really beautiful chymograph, which basically has a history of what actually happened in our different conditions on the DNA molecule. We think that this technology very often is not a standalone tool. It's more to complement your already existing life science toolkit. Many of you might already be using structural and compositional methods such as cryo-EM or also ChIP-seq. And this is, of course, amazing because it gives you incredible details of the structural and compositional details of your system, for example, of your molecular complex. But the problem is very often that it's a static picture, so you're kind of missing the dynamic information. On the other hand, many of you might already be using functional assays, such as live cell imaging. And these in vivo assays are, of course, very close to nature. However, it's very difficult to control the assembly of different complexes, or to actually control the external conditions of where this complex is happening, working. And then on the other hand, people, or you might already be using SPR or bulk assays to study biomolecular interactions. But this, on the other side, gives you typically average results, and the proof of a molecular mechanism can only be indirect. We indeed think that this dynamic single molecule analysis can bridge these two worlds, bridge structural and compositional information with functional information. Having introduced this technology as such, I would like to go into a couple of more real-world examples where this technology has been used to study different things, and we'll start with how this technology, this dynamic single molecule analysis, has been used to study gene editing errors. In gene editing, when people use, for example, CRISPR-Cas9 systems, it has always been a problem that you see a lot of off-target activity. So you want to target a certain gene, but somehow this complex is cutting somewhere else. And it has always not really been clear what's the reason for this off-target activity. And the one hypothesis in this field was that actually mechanical tension in the DNA leads to this off-target activity. And whenever mechanical tension on the single molecule level is involved, optical tweezers are a very nice tool to study these processes. What the authors of this paper then have done was they trapped two plastic particles into optical traps. They connected these two beads with a DNA molecule that has had a certain target sequence, and they incubated this with Cas9 complexes that had the corresponding guide RNA sequence. And then they were scanning along the DNA molecule and observing, okay, do we see on-target activity? Do we see off-target events? What's happening if we really expose this DNA to this Cas9 complex, which is firstly labeled, and how can we modulate that? And what happened there is really nicely visualized in this video that you see here. So you have these two optically trapped beads, which appear green here, and you have the DNA tethered between these two beads. You already see that at low force, you have this on-target Cas9 complex bound. Now, what you can do is you can pull the DNA and reduce mechanical tension. And indeed, you see that at higher forces, which are still biologically relevant, a lot of off-target binding happens. So that means that in the real world, in the cell, if you have this DNA under tension, for whatever reason, you increase the tendency of Cas9 to bind to very random positions on the DNA. You can now, of course, analyze these beautiful movies a bit further. So here is again one of these chymographs. And what you see here is now a sequence of different events. So it starts with having the DNA under low tension, so it's quite flexible, and you already see that you only have this on-target event. So Cas9 complex binds where it's supposed to bind. But now if you have this, if you stretch the DNA to 40 piconewton, which is again physiologically relevant, then you see a lot of off-target activity of this complex. You see it binding all over the place. Now you have this other part of the experiment where you relax the DNA again. You see unbinding of these off-target events, and only the on-target event stays. And this is very repeatable and reversible. And from locating these off-target events, you can of course also determine typical sequences where this Cas9 complex binds in case it does it performs off-target binding. And you can also use that to not only determine the sequence where it typically binds, but you can of course then also use this information to engineer your Cas9 complex and make sure that it has less off-target activity under mechanical tension. For other experiments, also the information of off and on rates of protein binding might be of relevance, but more regarding that later. And as I said before, we do not only have one color imaging, we have up to 3 colors, typically red, green, and blue. And now you can, of course, add up to 3 different components, 3 different differently labeled proteins, and see how they interact with each other. And then you would produce a video like that. So you have these 2 beads with the DNA in between, and you see different proteins that bind and unbind, and you can then kind of figure out if they co-localize, if they replace each other from the DNA, or whatever happens. The second application I would like to talk about is DNA repair. Of course, many of you might be studying proteins that are involved in DNA repair, and it would actually be really nice to observe how these DNA repair proteins actually act on the single DNA level, in the single DNA molecule level. And an example that has been performed in an actual real publication, I think it was published in Cell 2020, was the following. So there's a protein called RPA, which is a single-stranded DNA binding. And of course, in DNA damage, you very often have events where double-stranded DNA is transformed into single-stranded DNA. And we can model this transition from double to single-stranded DNA by pulling the DNA. If you exert a lot of tension to DNA, you open up so-called melting bubbles and you transition from double to single-stranded DNA. And we can now in real time observe how these repair proteins that recognize single-stranded DNA actually bind to this DNA molecule. Again, this chymograph, you have these two beads here, and you see that you can move one bead and use this to open up these melting bubbles. And if you do this in presence of this RPA, then you will see how indeed it in real time binds to this opened up DNA, and then also get replaced again if you close the DNA by relaxing it. And this is also reversible, so you can do it again and again. And of course, now you can, if you think a little bit further, you can expose these complexes of DNA and RPA to different downstream DNA repair proteins and see if, for example, they get recruited specifically where this initial protein already is bound. The third application I would like to talk about is genome organization. The genome is organized on many different levels. Of course, you have the DNA molecule, but this DNA is typically wrapped around histones, forming so-called nucleosomes, as you see here. And these nucleosomes then bind many more factors, which leads to higher order organization of this chromatin. And now a big question is, of course, how do these additional factors actually organize or in the first place bind to chromatin? And the study that I'm showing here was focusing on the protein PRC2, polycompressive complex 2. And the idea here was, okay, if we now incubate this chromatin fiber that is bound to these two optically trapped beads, can we detect how this additional protein, this PRC2, does actually influence the chromatin organization? And this is now also a nice introduction into a different kind of data that people typically generate using these optical traps. Before I was referring to videos and chymographs, but of course you also have all this mechanical information. And what people typically plot when they pull on something, pull on a DNA molecule, pull on a chromatin fiber, for example, is the following. So they plot the force here on the y-axis that you need to stretch this whole fiber to a certain extension. This is a so-called force extension or force distance curve. And the typical shape of these force extension curves gives you a lot of information about the structural transitions that happen in your chromatin fiber. So if you only pull on this chromatin fiber, that in that case, it was a 12-mer array. So you basically had a DNA molecule and 12 nucleosomes. You typically see these sawtooth-like patterns here. And each of these teeth is related to the unfolding or unwrapping of one of these nucleosomes. You can imagine you basically increase the force, and then at some point it ruptures, or this nucleosome gets unwrapped, and then the force drops and the extension increases. And now you can analyze these typical teeth structures here and get a certain transition force over the length difference plot in these transitions. And now you can, of course, do exactly the same with this chromatin organizing factor, in that case PRC2, and see how, if you have this PRC2 present, whether these structural transitions change. And what the authors then concluded from this data was actually the following. They observed that this PRC2 can bind directly to DNA, can bind to single histones, it can cross-link neighboring histones, and it can even cross-link non-neighboring histones. And this is, of course, very valuable information if you want to know how a certain factor contributes to DNA organization. The next applications I would like to talk about is about transcription and translation. The first example here is regarding the impact of nucleosomes on transcriptional dynamics. The assay that the authors set up here was the following. Now you had two optically trapped beads, and the DNA template was tethered to one bead, and on the other bead there was the polymerase II sitting. And this polymerase II was attached to this DNA template. And what now happens, typically is if your polymerase is active, it will walk along this additional part of the DNA that is not tethered, and it will start to transcribe it. And of course, it will run until the end. And if you keep this tether under a certain force, you can detect how the beads basically move away from each other. And this is exactly what is shown here. You can see, I mean, this position is basically a proxy for the length of the tether between the two beads. You see, while this polymerase is active, it's basically running until it stops. I mean, here it just reached the end of the DNA molecule. And this is a really nice direct readout of how different or how individual polymerase molecules transcribe DNA. Now, if you have nucleosomes here incorporated, this will happen differently. Now your polymerase will run until it encounters the first nucleosome. Then it will pause there and then slow down, and then it will keep running at some point once it skipped this nucleosome, and at some point encounter the second nucleosome. And you basically can, until it, well, reaches the end of the DNA. And this is, of course, a really nice example of how you can use optical tweezers to really study transcription on the single molecule level, and also observe the impact of different proteins that are bound to the DNA on the polymerase activity and transcription. The second example I would like to emphasize is related to translation. The idea here was now that ribosomes can open up RNA hairpin structures. And the question was a bit, okay, how do they do that? Do they need a certain cofactor? And the cofactor that the authors were studying here was EFG. And what they did here, they again held this whole tether. Here, the ribosome was directly connected to this hairpin and was supposed to go through it and open it step by step. And so they were keeping this under constant tension, and then were willing or aiming to observe again the stepwise opening by the ribosome action. And with that, this is of course accompanied by an extension or an increase of the extension between the bead to bead distance. And they combined it with fluorescence microscopy. So they were scanning with a fluorescence laser this region where the ribosome was attached. And this EFG was actually labeled fluorescently. So that means if whenever the EFG is present, you should, or is bound to the ribosome, you should see a signal. And this is exactly what they saw. So first of all, they saw that by monitoring the distance between the two beads, they saw that this ribosome is opening up the RNA hairpin in a stepwise manner. Stepwise increase in the distance between the two beads. And each step was actually accompanied by a fluorescence signal. So that means that in every step, EFG is binding to the ribosome. The ribosome is making a 3-nucleotide step in this through this hairpin, basically translating this hairpin, and then the EFG unbinds again until the next one binds, and we can go another step. And this is, of course, a really nice example of how this technology can be used to study such fundamental processes like transcription and translation on the single molecule level. And one thing I want to mention here is that both of these studies have not been performed with the C-Trap. They have been performed using home-built systems in these expert labs, and both labs are now actually using the C-Trap. That basically is reassuring us that we're on a very good way, and even labs that used to be home builders are now using our instrument. The next area of applications I would like to talk about is biomolecular condensates, which is a more and more popular field in biology. Maybe a little bit regarding the background. So cells have been found to be able to compartmentalize their interior even without lipid membranes by forming so-called biomolecular condensates. And they very often consist of intrinsically disordered proteins and nucleic acids. And these droplet-like structures have very interesting properties. They can form on demand, they can exchange constituents with the environment, and they have viscoelastic material properties. And they form in different areas of the cell, in the cytoplasm, for example, the Barbiani body or stress granules, and also in the nucleus, for example, by forming DNA damage foci or transcriptional bodies. And one study I would like to present is honestly my PhD work. And here we studied how actually single DNA molecules or single nucleic acid molecules can form condensates with certain proteins. The idea here was that, okay, can we observe this formation of condensates on the single molecule level? And which forces can be generated by such a process? And the assay that we used for that was relatively straightforward. We had these, again, 2 optically trapped beads with a DNA molecule in between. And we exposed this DNA to a protein that is typically involved in the formation of such condensates called FUS, fused sarcoma. And this was labeled with GFP. And what we did then was we relaxed the DNA and allowed it to condense. And what we were hoping to see was the formation of, indeed, a small condensate. And fortunately, that was exactly what happened. So you see these 2 beads and the DNA in between. This DNA is not directly labeled. What you see here is the signal of the fluorescently labeled protein, and you see how this protein homogeneously coats the DNA. It makes it basically sticky, and when you allow it to relax, you see the formation of the small blob. And at the same time, while observing this in the video, you can also measure the mechanical properties. So you can measure this force extension curve, for example, and compare it with naked DNA, and then infer things like condensation-free energies from that. The C-trap allows to study biomolecular condensates on many different levels, from macroscopic droplets or micrometer-sized droplets all the way down to the single molecule level. What people, for example, like to do is they like to trap these droplets directly. So now we don't trap plastic beads anymore, but we directly trap phase-separated protein and protein nucleic acid droplets. And you can try to fuse them and see directly if they are dynamic or if they just aggregated or whatever. Then you can also perform active microbiology. So you have a big droplet and you can put 2 small beads in there, wiggle with one, and then see how this wiggling is translated or transmitted through this droplet material. The third level of complexity that I want to show here is exactly what I've just shown before, the single molecule level, or well, let's say a single DNA molecule level, and many proteins. And you can see how individual DNA molecules, or probably also RNA molecules, can condense with proteins that have a tendency to condense. And if you want to go even into smaller structural level of detail, you can study individual proteins. You can have 2 optically trapped beads and a protein of interest in between, connected with 2 DNA tethers. And then you can directly pull this one protein. And as we know, you can, I mean, this is also what you've seen in this chromatin example. Now, from these structures that you see in these, while you try to unfold this protein, you can infer structural details of these proteins. And for example, you can, of course, compare different phosphorylation states of a protein and see if one that is associated to aggregation of proteins has a different internal structure than one that is less aggregation prone. And to make sure that you don't think that you can only study protein DNA interactions, I would also like to give an example of how people have used the C-trap to study force generation in cytoskeletal systems. Here, this is a study performed by our customers in Prague, Stanley Lansky and Marcus Brown, and they were interested in force generating processes that do not need active motors. What they have here is they have an active, or let's say a passive crosslinker called aniline, and they have actin rings, and they incubate these actin rings with this passive crosslinker. The crosslinker binds and basically constricts these rings. And this is, of course, a really nice example that you can also study using the C-trap. And what they did, they had, again, these actin bundles, incubated it with this passive crosslinker aniline, and on one hand, took really nice videos of when you have this fiber in between the 2 beads, you can pull it, and you see on one hand, this really nice co-localization of F-actin and also aniline. And then on the other hand, you can also really nicely measure the mechanical properties of these tethers or of these fibers by pulling on them. Having shown you not only the basics of the C-trap, of this combined optical tweezers with fluorescence microscopy, and having shown you a couple of examples where this technology can be used to study your biological system, I would also now say a couple of more words regarding LUMICKS and how we support you on your research journey. So we can help you with sample design and preparation. We on one hand have a very nice store where you can get, for example, biotin-related DNA molecules, DNA with certain fluorophores at certain positions, DNA with damaged sites. And we also have a nice biochemistry team that helps you to design your own samples. Secondly, we have people like me, application scientists, who can come over and help you with your experiments for a couple of days and also provide you with the required training. We support you with data analysis. We just released our new LakeView software, which is a really nice data viewer and a data exporting tool. And then, for example, we also have this Harbor platform where people really exchange scripts for experiment optimization and data analysis, and where people really, as a community, help each other to perform and analyze data. And lastly, something that is, of course, also provided by us, service and tech support. We have a really nice and quickly responding team of engineers that can help you to troubleshoot in the rare case the experiment or the instrument is not working as expected. And with that, I would like to thank you for attending, and I would like to encourage you to ask whatever you would like to know about the C-Trap. If you want to know more, visit our website or also contact me or Bartosz or Philip to learn more.